Welcome to this lecture on animal diversity. Today, we are going to explore one of the most monumental shifts in evolutionary history, the transition of life from water to dry land. Imagine trying to survive in a scorching desert without drying out. Amphibians struggled with this, but reptiles mastered it. We will uncover the brilliant biological engineering and terrestrial adaptations that allowed reptiles to break their ties with water and dominate terrestrial ecosystems. To understand how reptiles conquered land, let us first look at how they reproduce. Think about a normal frog egg. It is essentially a jelly-like bubble. If you leave it in the sun, it dries up like a raisin in minutes. Reptiles faced this exact problem when moving onto land. Their solution was an evolutionary masterpiece, the amniotic egg. By creating a specialized shelled egg, they essentially invented a way to carry the ocean with them. This portable pond provided a safe, enclosed environment, drastically increasing offspring survival and changing the course of animal history forever. Now that we know why the amniotic egg is so important, let us examine its internal structure. The egg is not just a shell. It contains 4 distinct extraembryonic membranes. First, the amnion acts like a private indoor pool, cushioning the embryo and preventing it from drying out. Second, the chorion works like a lung, allowing the baby reptile to breathe through the shell. Third, the yolk sac is the packed lunch, providing long-lasting nutrients. Finally, the allantois acts like a trash can, safely storing toxic waste. Together, these structures make independent development on land possible. Understanding the structure of the egg leads us to a fascinating physical challenge. How does the embryo breathe inside a sealed shell? The answer lies in microscopic pores, governed by Fick's law of diffusion. We can model this using the gas conductance formula. Here, G represents the gas conductance, or how fast oxygen enters. This depends on A, the total area of the pores, multiplied by D, the diffusion coefficient, and delta P, the pressure difference driving the gas inward. We then divide all of this by L, the shell thickness. Physically, if the shell becomes thicker, conductance decreases, making it harder to breathe. If the pore area increases, more oxygen flows in, allowing faster growth. Before we move on, let us see this formula in action with a concrete example. Imagine a desert tortoise egg. We are given a shell thickness of 0.05 centimeters, a pore area of 0.02, a diffusion coefficient of 0.2, and a pressure difference of 10 units. We need to find the gas conductance. First, we substitute these exact values into our formula. Next, we multiply the numerator values together, giving us 0.04. This represents the raw potential for oxygen flow. Finally, dividing by the shell's physical resistance, we get a final answer of 0.8 cubic centimeters per second. This means the embryo receives exactly enough oxygen to survive without losing too much water. Now that we have calculated how the egg breathes, let us look at a visual representation of this incredible structure. On screen, you see a cross-section of the amniotic egg. Notice the tough, leathery outer shell that provides physical protection. Just inside, the chorion lines the shell, and you can see the red arrows representing oxygen entering and blue arrows showing carbon dioxide exiting through the microscopic pores we just calculated. In the center, the embryo rests safely inside the blue, fluid-filled amnion. Below it is the large yolk sac providing food, and to the side is the allantois collecting waste. This diagram perfectly illustrates how the egg functions as a complete self-sustaining life support system. Having solved the problem of reproduction on land, reptiles still faced the threat of drying out themselves. Think about wearing a wet cotton shirt on a hot, windy day. The moisture evaporates, leaving you dry. Amphibians have skin just like that wet shirt. If they stay in the sun, they perish. Reptiles, however, evolved the biological equivalent of a heavy-duty waterproof raincoat. They developed a heavily keratinized epidermis that forms thick, overlapping scales. Unlike frogs, reptiles completely lack mucus glands, making their exterior dry and highly impermeable. This waterproof integument locks moisture inside, allowing them to bask on hot rocks and hunt in the open sun without turning into dried husks. Beyond their waterproof skin, reptiles also had to change how they process waste to conserve water internally. Most animals, including humans and amphibians, flush out toxic nitrogenous waste using large amounts of liquid water. In a desert, this is a deadly waste of resources. To solve this, reptiles became uricotelic. This means they convert toxic waste from protein metabolism into uric acid instead of urea or ammonia. Because uric acid is highly insoluble in water, it does not need to be flushed out. Instead, it is excreted as a dry, semi-solid white paste. This brilliant internal adaptation saves every precious drop of water, allowing desert reptiles to survive entirely on the trace moisture they get from eating their prey. To truly appreciate these adaptations, we can mathematically model how well a reptile retains water compared to an amphibian. This brings us to the evaporative water loss, or EWL formula. Here, EWL represents the total water lost per hour. This is calculated by multiplying K, the skin permeability constant, by SA, the total surface area of the animal. We then multiply that by the difference between C in, the water vapor concentration inside the animal, and C out, the concentration in the dry air. Physically, this tells us that water loss is driven by the dryness of the environment, but is heavily restricted by the skin's permeability. If the permeability constant K decreases, the total water loss drops significantly. Let us apply this formula to see exactly why a frog cannot survive in the desert. We will calculate the evaporative water loss for a frog with a surface area of 50 square centimeters. The concentration difference between its moist body and the dry air is 20 units. Because the frog has wet, glandular skin, its permeability constant is high, at 0.5. We substitute these values into our equation. Multiplying 0.5 by 50 and then by 20 gives us the total water lost through the skin. The final answer is a massive 500 units of water lost per hour. This extreme rate of dehydration explains why frogs are permanently chained to moist environments. Now, let us perform the exact same calculation for a reptile to see the impact of its adaptations. We take a lizard with the exact same surface area of 50 square centimeters, facing the exact same environmental dryness of 20 units. However, because of its thick, keratinized scales, the lizard's permeability constant is incredibly low, at just 0.002. We substitute this new permeability into the formula while keeping everything else identical for a fair comparison. Multiplying 0.02 by 50 and then by 20, we find the lizard's water loss. The final answer is only 20 units of water per hour. This dramatic reduction showcases the sheer power of reptilian skin. Having calculated the water loss for both animals, we can now mathematically prove the exact survival advantage granted by the reptile's scales. This chunk of our proof sets out to compare the two species directly. To do this, we set up a mathematical ratio, dividing the evaporative water loss of the frog by the evaporative water loss of the lizard. By substituting the final values we just calculated, we get a ratio of 500 divided by 20. This step is crucial because it isolates the effect of the skin permeability, allowing us to quantify exactly how much better the reptile is at holding on to its internal water compared to its amphibian ancestor. Continuing with our proof, we now simplify the fraction to find the final result. Dividing 500 by 20 gives us exactly 25. This means the evaporative water loss of the frog is 25 times greater than that of the lizard. Or conversely, the reptile loses 25 times less water under the exact same harsh environmental conditions. This mathematical proof perfectly illustrates the intuition behind terrestrial adaptation. A single evolutionary change in skin permeability creates a massive biological advantage. This incredible efficiency is the undeniable secret to reptilian dominance in arid ecosystems, proving why they can thrive where amphibians would quickly perish. To make this mathematical proof even clearer, let us look at a visual comparison. On screen is a bar graph plotting the evaporative water loss we just calculated. The vertical axis represents the water lost per hour. Look at the massive blue bar on the left, representing the frog. It shoots all the way up to 500 units. Now, compare that to the tiny tan bar on the right, representing the lizard, sitting at just 20 units. The arrow highlights this staggering 25-fold difference. This diagram visually reinforces what we proved mathematically. The keratinized skin acts as an ultimate shield against the drying power of the sun, making life in the desert possible. We have covered a lot of ground today, so let us summarize the key takeaways. First, we learned that the amniotic egg was a revolutionary invention, acting as a portable pond that completely freed reptiles from needing water to reproduce. Second, we saw how its specialized membranes perfectly balance the need to breathe with the need to retain moisture. Third, we proved mathematically that thick, keratinized skin drastically reduces evaporative water loss compared to amphibians. Finally, shifting to urocotelic excretion allowed reptiles to save internal water by producing solid waste. Together, these brilliant adaptations engineered reptiles to become true masters of the land. You now understand the fundamental biology of terrestrial survival.